Hello, everyone. Thank you for joining us today for our second genetic counseling webinar. My name is Colette Royal. I am the lab director here at McKinley Caregiver Resource Center. Today, I will be introducing Sharika Amin. She is a second-year genetic counseling student at Emory University School of Medicine Genetic Counseling Training Program. She obtained her undergraduate degree in biotechnology from the University of Mumbai, India. In India, she worked at a cancer genetics clinic for a year before coming to Atlanta at Emory. She is currently pursuing her capstone resource project at Emory ALS Center on the experiences of patients with genetic testing for ALS and is very interested in genetics of neurological conditions. Sharika, it's all yours.
Thank you. Thank you for having me. Okay. Okay, I should be able to share my screen now. Let me know when you can see my slides. Or could you?
Yep.
Are you able to see my slides?
Yes.
Okay, wait, I'm not able to see them yet. Okay, great. Great, so thank you so much for the introduction. And like Ms. Royal said, I'm going to be speaking about the genetics of neurological conditions. And yeah, and if anyone ever has any questions, I guess you could feel free to put it in the question and answer section of the chat, and I'll be happy to answer them maybe during the session or after it. Great. So the things that I want to cover today are basically the basics of genetics and what genetic testing is, yeah, basically what genetic testing is, go over the genetics of neurological conditions, mainly focusing on ALS, Parkinson's, and Alzheimer's disease, which I believe is what most people were interested in. And then finally, what the implications of genetic testing are. So things to keep in mind when you're pursuing genetic testing and really how testing affected people versus testing at-risk family members is different. Great, let's get started. So really the basics of genetics, it's really complex, and I'm going to try to make it as easy and digestible as possible for us to understand. When we're thinking about what a gene is or what DNA is, it's easy to think about how, our whole body is made up of cells, and each cell has now can you all see my cursor on the screen? Okay.
Yes.
So our whole body is made up of cells, and each cell has a nucleus in the center, and in that nucleus is our DNA, right? This DNA is packaged as chromosomes, and all of us have 23 pairs of these chromosomes, right? One part of each pair we get from mom, one part of each pair we get from dad. And so this DNA is basically just a really long sequence of letters. So like how the English alphabet has 26 letters, the language of DNA has 4 letters, A, T, C, and G. And DNA is just a really long sequence of these 4 letters. So just billions and billions of these letters in a really long sequence and how we like to think of it is that we break up this really long sequence into smaller sections called genes. And each gene has its own function, right? Each gene basically makes instructions for things called proteins, and these proteins all have different functions. So there's genes that code for proteins that decide eye color, hair color, all the different functions of our body. There's genes for those things, and those genes code for these proteins. Great. And so if there's any change in these genes, so if there's like a big part that's missing, or if there's one spelling mistake in the sequence of the genes, it could really affect how this protein is formed, and that's when we have certain conditions that could develop depending on what the gene's original job was. So there's, like I said, there's genes that decide different things. There are genes that decide how our brain works and how nerves fire up and how nerves do their job. So if there's something wrong in the instructions for those proteins, that's when people would be at the risk of developing certain neurological conditions. Another way to think about it, just to make it easier, is if we think of all of our DNA as a library of 23 bookshelves. So each bookshelf would be one chromosome, and each chromosome has multiple genes on it, and you can think of each gene as one book. And then the book, like I said, it will be written in DNA's language of those 4 letters, A, T, C, and G, and the books have the instructions for all these proteins. If there's a spelling mistake in the book, or if there's a few chapters that have gone missing, or a few chapters that have doubled up, anything that goes wrong in the instructions of creating the proteins really leads to genetic conditions. But I thought that's a really interesting analogy that people are able to keep up with more than the genetics. Right. So basically with genetic testing, what we're doing is we're looking for damaging changes. And these damaging changes are commonly called mutations. So like I said, if there's, instead of a C, there was supposed to be a G, and that really messes up the instructions. That's what we're looking for. We're looking for these changes that are damaging in the DNA that could lead to these conditions. So we read through the sequence of the DNA. So another important thing to keep in mind is that there's different kinds of genes that could be associated with conditions. So there's some genes, when there's a change in them, they directly cause the condition. So we know if someone has a mutation in one of these causative genes, they will have the condition at hand. But there's other genes that we think of as susceptibility or risk factor genes, which is when if you have a mutation in them, it only increases your risk of having the condition. It does not mean that you will develop the condition in your lifetime. And this is a really important factor, especially in neurological conditions, because while there are causative genes, there are a lot of risk factor genes for neurological conditions, which can get really tricky when we're just testing for them or telling someone that they're at risk of developing neurological conditions, because that's a little bit harder to digest or think about. But we will get into that a little bit later. So for most neurological conditions, there's something called autosomal dominant inheritance. And that's basically, like I said, we get 50% of our DNA from mom, 50% of DNA from dad. Oops. And so we all have 2 copies of each gene. And with autosomal dominant conditions, only one of those copies needs to have a mutation or a damaging change in it for us to have the condition. So let's say there's 2 parents who want to have a child or already have children. Every time the person has a child, you only pass down one of your copies. You don't pass down both of your copies to your children. So every time this person had a child, they could have either passed down the working copy to the child, or they could have passed down the copy that has the mutation on the child to the child. So that leaves, so for every child that they had, the child could have a 50% chance of inheriting the mutation and 50% chance of not inheriting the mutation. And the only way we would know if the child inherited it was by performing genetic testing for the child. And like the diagram says here, the risk of inheritance is the same for females and males. It does not depend on gender for autosomal dominant conditions. But having said that, most neurological conditions are multifactorial, and what that means is that there's multiple factors that decide whether a person is going to develop a condition over their lifetime, right? And that's just like conditions like diabetes and other common conditions that we like to think of, like heart disease. Those also have multifactorial inheritance. And to make this concept a little bit more easy to think about, I really like this diagram. So if we think of this jar, we think that everyone has a certain threshold that they have to meet to develop a condition, and there could be multiple factors that contribute to people reaching this threshold. So some factors are genetic factors, and some factors are environmental factors like your lifestyle choices or or just like exposures that you've had in your lifetime to like chemicals and things like that. And there's also unknown factors. There's a lot of things that we don't know how they contribute towards our likelihood of developing a disease also. So there's multiple factors. So how we would like to think of it is that a person is born with certain genetic factors, but over their lifetime, they keep accumulating these environmental factors, and at a certain point in their lifetime, over time, they've accumulated enough environmental factors for them to reach that threshold and to develop a condition, right? And so if someone so like if we think of someone who was born with more of these genetic factors, it would take lesser time and lesser environmental factors for them to reach the threshold of developing the condition. But then there's also another thing to keep in mind is that there's some conditions that really all it takes is the genetic factors, like Huntington's disease. If we think about it, really people who have the genetic factors for Huntington's disease will develop Huntington's disease in their lifetime, and those environmental factors we don't think contribute as much, but conditions like Parkinson's or Alzheimer's, really, there are these other environmental factors do play a role as well. So starting off with the conditions, I'm going to start with ALS, or amyotrophic lateral sclerosis. It's also commonly called Lou Gehrig's disease. It is a condition that affects parts of the nervous system that control voluntary muscle movements. So the muscles that we move at will, like our arms and legs, more commonly. And these motor neurons are basically the line of communication between your brain and these voluntary muscles. So people with ALS usually start showing symptoms with their head or neck, so like impaired speech, having excess saliva, or difficulty swallowing, or they start showing symptoms with their lower body, so like with their issues with their legs. So tripping and falling downstairs and having weak feet and the inability to walk. This condition really is progressive, so all these symptoms become worse over time, but yeah, they become worse over time. And then, like I said, there's multiple of these symptoms really limit their ability to live an independent life, like being able to dress themselves or being able to prepare food for themselves. And they have a limited range of motion after some time. And they gradually lose strength in their muscles and become weaker, and it also becomes hard for them to breathe. So it really is a hard condition to live with, but even though it's progressive, how it progresses differently in different people. Some people, it progresses in fast, like over the course of 2 years, they would develop most of these symptoms, and some people over the course of 10 to 15 years would develop these symptoms. Currently, there's no cure for ALS, but there are multiple clinical trials in the works for finding a cure. There is medication available to slow down the progression, to slow these symptoms down, but really nothing to completely stop them. So coming to the genes associated with ALS. So when we think about ALS, if we conceptualize it as, if we think of 100, if we take 100 people who have ALS in a room, 10 of those people will have familial ALS. And what familial ALS is it means that there's multiple people in their family who either had ALS or conditions related to ALS. And 90 of the remaining 90 in the room would have had sporadic ALS, which means ALS that just occurred by chance or for many of those unknown reasons that we were talking about before. So I like this diagram because it puts it into perspective. So like I said, 10% of those people with ALS have familial ALS and 90% have sporadic ALS. This diagram also tells us all of the genes that they know are associated, not all, some of the genes that are associated with familial ALS. The big gene that's associated with ALS is called C9orf72, but I will refer to it as C9 for the sake of time. The C9 gene, as you can see, for people with around 40% of people with familial ALS have a mutation or a change in this C9 gene. The other genes are SOD1 and a few other genes. But as you can see, there's still 20% of people who have a family history of ALS and related conditions, but we're not able to find exactly which genes, which specific genes these changes are in. So there's still a lot we don't know in the genetics of ALS. And these genes that we're talking about now were also pretty recently discovered, with C9 being discovered only in 2011. So there's still a lot that we're learning right now about the genetics of ALS. And that's also really important to keep in mind when we're offering testing to patients. But another interesting thing is that even for people who do not have a family history of ALS, these sporadic cases, still around 6 to 7% of those people will have a mutation in the C9 gene. So really, genetic testing isn't only indicated in people with a family history, but really anyone with ALS. But like I mentioned before, for sporadic cases, the causes largely remain unknown, or are those environmental factors that we cannot quantify right now. I wanted to talk a little bit more about C9, just because it really makes up a majority of people who have familial ALS. It is a little different from the other genes associated with ALS, in that these other genes like SOD1 and FUS, it's really those spelling mistakes that we were talking about before that cause the condition. But with C9, it's basically, if we go back to our analogy of books, in the book of C9, there's a section where there's these 6 letters, G, 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 C, C, which are usually repeated again and again. And if they're repeated up to 30 times, it's usually normal. We expect them to be, expect there to be 30 repeats of these 6 letters in someone's DNA. But if it's over 30 repeats is when they would have ALS or frontotemporal dementia. And that's another unique thing about C9. It doesn't only cause ALS, it's also known to cause frontotemporal dementia. And what's interesting is it's really a spectrum. So someone who has more than 30 repeats in C9 could have only ALS, they could have ALS and frontotemporal dementia, or only frontotemporal dementia. And we don't know enough about this gene for us to know when and how they would have either of these. Like, would they have only ALS, only frontotemporal dementia, or both? So we don't know. The genetics of it is still not clear for us to know how the genes decide which of these are, where on the spectrum someone would fall. But we know that for someone who has more than 30 repeats in this C9 gene, 50% of those people would be affected with either ALS or frontotemporal dementia by the age of 60, and 100% of them would have either ALS or frontotemporal dementia by the age of 80. So that's really important to keep in mind. Okay, so the next condition I wanted to talk about is Alzheimer's disease. Alzheimer's is the most common cause of dementia, and it affects memory, thinking, and behavior. Just like ALS, it is also progressive, so it gets worse over time. And it gets severe enough to interfere with people's daily life and daily tasks. There's usually an average course of 8 to 10 years for someone with Alzheimer's, but that really can differ from person to person. There is treatment available for symptoms. So there is treatment to slow down the symptoms and improve quality of life, but just like ALS, there's no cure. An interesting fact about Alzheimer's is that it is really difficult to diagnose. Most confirmed diagnoses are usually done only after death, where we can look at the brain and we can actually see the things that point out Alzheimer's in the brain. But the CDC has reported that most people living with Alzheimer's either don't know that they have Alzheimer's or have been told, and neither them or their caregiver don't know that they have Alzheimer's. So there's really a lot of people out there who haven't been diagnosed. A major risk factor for Alzheimer's, though, we know is old age. So as someone gets older, their risk of developing Alzheimer's increases. So when we think of Alzheimer's, we think of 2 types of Alzheimer's: early onset and late onset Alzheimer's. Early onset is basically below the age of 60 to 65 years old if someone is diagnosed, but this makes up really the minority of people with Alzheimer's. Only 1 to 5% of people with Alzheimer's are diagnosed before the age of 60 to 65. And that could mainly be because, like I said, the major risk factor is age. But more commonly, familial Alzheimer's, early onset Alzheimer's is more commonly familial. And like by familial, we mean that there is a family history of Alzheimer's, right? So like how in ALS, it was a 90% versus 10% of people who have familial ALS. In Alzheimer's, about 25% of people with Alzheimer's have familial Alzheimer's. So it's a little bit more over here. And then for late onset Alzheimer's, like I said, it's most people with Alzheimer's have late onset over the age of 60 to 65. This is a pedigree. This is how we document family history. So to give you a little bit overview, I know it's a little hazy and we can't see it too well. I'm sorry for that. But squares are males and circles are females. Anyone who's red in color is someone who's affected, and someone who's blue is not affected by Alzheimer's. People who are connected by horizontal lines are partners or have had children together, and people connected by vertical lines are children. So these are parents, these are their four children, and then those are the children's partners and their children. But this is what we would expect someone with familial Alzheimer's family pedigree to look like, or if there's a known genetic cause of Alzheimer's in the family. So getting to that, what are the genes associated with Alzheimer's? So for people who have early onset Alzheimer's, these 1 to 5% of people, there's genes that are inherited in an autosomal dominant manner. The first gene is called APP. And by complete penetrance, it just means that anyone who has a mutation in this APP gene will develop Alzheimer's in their lifetime if they live up to the age of around 75 to 80 again. And the same goes for PSEN1. Anyone who has a mutation in this gene will also develop Alzheimer's in their lifetime. And then PSEN2 is 95% of people with mutations in this gene will develop Alzheimer's, whereas 5% of people with mutations in this gene will not develop Alzheimer's. And these are causative genes. So remember in the beginning how we were talking about how there's causative genes versus risk factor genes. These are genes that we know cause Alzheimer's and early onset Alzheimer's. But there is a risk factor gene associated with Alzheimer's called ApoE, which I'm going to talk a little bit more about later as well. But ApoE is really important to remember is a risk factor gene and not a susceptibility gene. So if someone has some specific changes in the ApoE gene, it does not mean it's not sufficient for them to have it's not sufficient enough to cause Alzheimer's disease. So really clinical testing of ApoE isn't performed because there's no clinical utility to performing the test. There's nothing nothing that's recommended off of testing for ApoE. And the American College of Medical Genetics and Genomics and the National Society of Genetic Counselors recommend against testing for ApoE for patients without any symptoms, especially. Great. And the third and last condition that I want to talk about is Parkinson's disease. It is the second most common neurodegenerative disease, and it affects the neurons in our brain that produce dopamine. It is again a progressive and adult onset condition, just like the other two most of the time. And the symptoms include tremors, difficulty balancing, bradykinesia, which is basically slow movements, stiffness in the muscles, and disturbances in walking, which is the gait and posture. Similar to ALS, around 10 to 15% of Parkinson's disease is familial or genetic. But again, Parkinson's disease is a good mixture of your genes with environment and your lifestyle, not just your genes. It's the multifactorial inheritance that we were talking about before. And there's different types depending on the onset. So there are people who have Parkinson's disease before the age of 20, and that's juvenile onset. And usually when it's juvenile onset, it's genetic, because like I said, if you're born with more of those genetic factors, it takes lesser time for those environmental factors to stack up and reach that threshold. So juvenile onset is more often than not genetic. And then there's early onset and late onset, which is about the age of 50. Age is again the largest risk factor for Parkinson's disease. So most people with Parkinson's have late onset Parkinson's disease. And there is no cure again, but there's medication to manage these symptoms. There are, however, clinical trials available for people to enroll in. Genetic testing isn't routinely performed for people with Parkinson's disease because, again, it's something that we're still learning more and more about every day, but and hopefully will become part of people's care in the future. And some environmental factors that that predispose people to Parkinson's is head injury. There could be some things related to the area that they live in, their occupation, exposure to metals and chemicals, herbicides and pesticides also are some environmental factors that lead to Parkinson's disease. Great. And when we're thinking about genes associated with Parkinson's disease, there are multiple. This graph is a good way to look at it, because so these are the genes that we know are causative. So again, those causative and risk factor things that we were talking about. These genes on the top, we know that if people have mutations in them, those mutations would cause Parkinson's disease. But then there's these genes at the bottom, which are risk factors for Parkinson's disease, where it only increases someone's risk of developing Parkinson's and doesn't always cause Parkinson's disease. But another interesting thing about some of these genes is that not all of them are inherited in that autosomal dominant manner that we were talking about before. Some of them are inherited in an autosomal recessive manner. And what that means is that for someone to be affected, they need to have both of their copies of that gene mutated and not just one, like in autosomal dominant. So both of the parents of that person would have been a carrier for that gene, and every time they had a child, they could have either passed down the normal copy or that copy with the mutation, but the person will be affected only if both the parents pass down the copy with the mutation. So that's a 1 in 4 chance that the person would be affected if both the parents were carriers for a mutation in some of these autosomal recessive genes. And some of these autosomal recessive genes are this pink one, PARK2, and ATP1382. These are genes that are inherited in this autosomal recessive manner. And these autosomal recessive genes are the ones that usually lead to juvenile onset Parkinson's disease. So Parkinson's disease seen below the age of 20 years old. Some of these genes like GBA also cause other conditions when they're inherited in a recessive. GBA causes Parkinson's disease in an autosomal dominant manner, but if someone has both of their copies of GBA mutated, they would have another condition, a lysosomal storage disease condition. So there's multiple, the takeaway should just be that there's multiple genes associated with Parkinson's disease having different forms of inheritance. A lot of them are risk factors and are seen more commonly in the general population. But an interesting thing could be that if you have enough of these smaller risk factors, maybe your risk is as high as someone who has one of these big risk factors. And that's something that we don't really do clinically yet, is look at these smaller risk factors to see how they add up to dictate someone's risk of Parkinson's disease. Right now, testing would only be offered for these genes that cause Parkinson's or are high risk, but not so much for these low risk genes that are seen more commonly in the population. Great. So that was a lot of information about the different types of neurological diseases and different genes associated with them and all the different inheritance patterns. So genetics is really complicated, and to complicate it more, it's not like usual medical tests where we have just a positive or a negative result. So usually we do genetic testing on a blood or a saliva sample. When we do medical tests, we usually expect black and white answers of positive or negative, but that's not always the case with genetics. But we can start off with, so if you undergo genetic testing, one of the possible results is a positive result. And what that means is that we found a damaging change in a gene that we know is associated with this condition that you're getting tested for. And that's when we can recommend genetic counseling and testing for family members, because we would know how that gene is inherited, and we would, know the risk of passing on the mutation to the next generation. Like for autosomal dominant conditions, if someone's mother has the condition, we would expect that the child has a 50% chance of having the same mutation or 50% chance of not having the same mutation. And we would then recommend testing for their children or their siblings or their parents. Having a positive result can confirm a diagnosis for someone. So like I said, with Alzheimer's, it's so difficult to diagnose someone before they pass away without taking a look at their brain. But if they had a mutation in one of those causative genes, it could confirm a diagnosis of Alzheimer's for them. Or it could identify the risk of developing a condition in the future for those family members who don't have the condition yet. The second type of result is a negative result. That's when there's no damaging changes found in the genes tested, and everything looks like it's supposed to look. This does not change the diagnosis for people with symptoms. So if you already have symptoms of, let's say, Alzheimer's, or if you already have a clinical diagnosis of Alzheimer's, a negative genetic test result does not take away that diagnosis. But it can indicate that the person does not have an increased risk of developing a certain condition at that point in time, because the test could have missed a damaging change that leads to the condition. Because like I said, there's so much unknown in all of these neurological conditions. So we could have maybe it's the changes in a gene that we haven't discovered yet, or it's the changes in a gene that wasn't tested for in this particular test. So there could be some residual risks yet for people who test negative, because we don't know all the genes associated with these conditions. And then the last kind of result is this gray area between positive and negative, which is called a variant of uncertain significance, which is basically when we find a change in someone's DNA. So we find there is a change, but we're not quite sure if that change is disease-causing or not disease-causing yet, because there's just not enough research done on that specific change in the DNA. Right, so we don't know if it's disease causing or it's just a change that makes us look different and does not affect health. So we all have certain variation in our genes that makes us look different, or makes us work differently than other people, right? So we don't know if this change that we have found could cause disease or it does not affect health at all. So usually when there's a variant of uncertain significance reported, the lab will continue to do research to reclassify it into a positive or a negative result eventually, and will recontact you when that is done. Okay, so those are the three kinds of results. Another place where genetic counselors are really helpful and are required really is because genetic testing options are really complicated and there's multiple options. So when someone is undergoing testing clinically, you could either do single gene testing. So you could test only one gene. So for example, in ALS, maybe if someone has a history of ALS and frontotemporal dementia, so you would instantly think the C9 gene is implicated here. So you want to start by testing only the C9 gene, and then if that comes back negative, go and test all of those genes that we know are associated with ALS. So then go to a gene panel. So there's the option of testing just one gene or testing multiple genes that we know are associated with all of these conditions. And that really works on an individual basis of how much information the patient wants, or depending on the family history, we would decide whether a single gene or a gene panel test is required. And really, when we're looking, an important thing to keep in mind is the more genes we look at, really that uncertain result becomes more likely, because just we're casting a wider net, and there's more chance of getting one of those uncertain results if we're looking at more genes. And that's one of the things that we like to explain to patients before we order testing. And then the second kind of testing that there is, like I was talking about ApoE before with Alzheimer's, it's a risk factor for Alzheimer's. If someone has a mutation in the ApoE gene, it doesn't necessarily mean that it's not enough to cause Alzheimer's for them, but there are testing laboratories that offer direct-to-consumer testing, so not through a provider, for ApoE. And ApoE isn't clinically recommended by all of our professional societies. So it's really, we have to be a little bit wary about like getting tested for it direct-to-consumer. And my classmate Jay is going to come soon, I know, to talk more about it. So I'm not going to go too much into the details of it, but just know that it's not clinically recommended to perform those kinds of tests. So genetic testing is really a complicated process because there's just so many implications of undergoing testing, and I want to touch on a lot of them because that's the reason we require so much genetic counseling and so much information giving to patients before they undergo testing, just because it can impact their life in multiple ways. So some of the benefits of undergoing genetic testing include obviously providing a sense of relief from uncertainty. There's a lot of people who don't know that why they've had this condition going on in their family for so long, or they've had, it's hard for them to get a diagnosis, and it's a relief almost for them to have a reason for this condition going down in their family. It helps people make informed decisions for the future, so about healthcare, about what kind of life insurance they should get for the future, or how they should organize their finances to keep away for more treatment or for clinical trials, or how they would want to have their lifestyle depending on their genetic risk already. Right? Some people with ALS, for example, if they find out early enough that they have this predisposition to develop ALS, they like to make some lifestyle choices, like buying a house with just one floor so it's easy for them to access everything in the house and things like that. So it really can give you anticipatory guidance. So really tell you how to prepare for the future if you want that information. Yeah, so basically it can provide prognosis information about like what kind of, so if someone is unaffected at the moment and they have for example, a mutation in that C9 gene, eventually in their life they know they're going to develop either ALS or FTD if they live up to the age of 80. And then a lot of clinical trials need a genetic test report for enrollment criteria, because there's a lot of clinical trials not specific to genes, especially in ALS, and Parkinson's disease and Alzheimer's disease are catching up with that. And knowing a specific change running in the family can help people make reproductive decisions about how they want to have children or when they want to have children. There are methods for us to prevent the next generation from inheriting a mutation that the parents have. So there's reproductive genetic counselors that would be available to discuss those options of how to prevent the next generation from inheriting a disease-causing mutation. And this really just clarifies risk for family members. So if an affected person tests positive, like I said, we can know who to test in the family. Those are some of the benefits of genetic testing. When we're thinking about limitations of genetic testing, genetic testing is an emotional process. It can be it can be socially difficult for you to explain this to people in your life about the consequences of genetic testing. There could be multiple financial consequences of genetic testing as well. If someone has seen their parents or their siblings go through this condition, it's really hard for them to process having to go through this condition themselves. A lot of people are more often than not concerned about their children than they're concerned about their own risk of developing the disease. But it really is an emotional process for people to think about developing any of these neurological conditions just because they impair your lifestyle so much. There's also issues of family tension, because there could be some family members that want testing, some family members that decide they don't want testing, and it's just really, it can be hard to talk to your family about this risk of developing a condition, especially if you all, as a family, don't usually talk about health concerns or don't discuss health concerns, or you want to reach out to your first cousin to let them know you found out about this mutation in your family. It can be a lot of complex things to think about, and that's really where your genetic counselor steps in and helps you think through this process of how are you going to communicate this information, how are you going to talk to this particular family member about it, do you need me to write a letter for you to give this family member. So it can lead to family tension as well. There's concern for genetic discrimination, and what that means is that so there's this law called GINA, Genetic Information Non-Discrimination Act, which protects individuals from being discriminated against according to their genetics by their employers and by health insurance. So your employer cannot change your salary, or your health insurance cannot change your premium or anything based off of a genetic test result. But this law does not apply to life insurance, disability insurance, long-term care, and active military personnel. So really, there could be, so life insurance could potentially discriminate against you based off of your genetic test result and increase your premium. So before someone who is unaffected gets tested for these conditions, we usually recommend that if they want life insurance or long-term care, they get it in place before they pursue genetic testing. And then there's the limitation of how genetic testing can only provide limited information currently, because like I said, we don't know everything. We don't know all of the causes or all of the genetic causes of any of these conditions, so we could be missing out on things just yet. Another limitation is that there's no cure for most of these neurological disorders. So you could undergo testing only to find out that you're at risk of developing this condition in the future, but there's no cure for this condition yet. And cost and insurance coverage of testing really is a controversial topic with genetic testing, because really some insurances cover genetic testing, some insurances don't cover genetic testing, and your genetic counselor should be able to navigate that and help you figure out the path of least resistance over there. One of the last things that I wanted to touch upon was how testing an affected person is different from testing a person who is not affected or an at-risk family member. So both of them have like psychosocial implications, right? It's a hard test to go through regardless of whether you're affected or not. If you're affected and you have it, then there's a risk of your family members having it as well. And those people deal with a lot of guilt that they pass it on to their children. But if you're unaffected, you're dealing with the consequence of, if I come back positive, I might develop this condition. I don't know when I will develop the condition. You're going to be living with that. So that's something that's hard to live with as well. So like I said, so for these at-risk people, we cannot predict the onset of symptoms or the age of onset or how the disease will progress. So it's really difficult for them to process that information that at some point in their lifetime, they may or may not develop this condition. Yeah, and like I said, so some damaging changes or some mutations don't always lead to a condition. So you're really sitting on this rock of uncertainty. And there's no preventative treatment. So there's nothing that anyone who comes back positive can do to prevent themselves from having this condition, which can be hard for some people to deal with because a lot of people are doers and they want to do something. But luckily for some of them, especially in ALS, there's trials for unaffected people who have genetic changes. Again, that might become more common with other conditions in the future as well. So really, these people, both affected and at-risk individuals should consult their neurologist and genetic counselor about all of these different implications. And there are guidelines, especially I know for ALS and Alzheimer's, for pre-test counseling. So before you get the test, you should really we offer people to bring a support person with them, just because we know this is hard to talk about. We discuss genetic discrimination. We discuss genetic testing as an option. We never say that you have to do it, because it really is a personal choice about how much information you want and whether you want to undergo testing and find this out. Timing can be a big deal as well. Some people don't want to get tested right now. Maybe you just gotten married. Maybe it's a really difficult time in your life. Maybe it's holiday season. So really timing can be a big factor there as well. So we explore the reason as to why people want to undergo testing and what they think they can do with the genetic test result. Discuss how and when they want the result, like if maybe they want to get tested now but want the result after a couple of years and not now. So really we discuss all of these things with them. We want to inform them that there's no current cure, but there are options for clinical trials for some of these conditions, and keep following up with these people even after we give them the genetic test result, to keep them updated with all the new things that are coming up for their condition. So the last thing I have is just us running through a few scenarios. Like I said, this is a pedigree. Same thing goes, squares are male, circles are females. Anyone who's colored in, let's say, has a history of ALS, because this is the ALS family pedigree. Let's say that the person with the arrow is your patient right now, and her mom has ALS and her brother has also had ALS. We usually prefer if an affected family member gets tested before an unaffected family member, because that's when we could really get a true negative result for this person. And I'll explain what that means. So let's say this person comes in, but we say that her brother and her mother are ideal candidates for genetic testing, and her mom undergoes testing and comes back positive for a mutation in SOD1, which causes ALS, like we know. So in this case, then this person can then undergo testing, because right now her risk of inheriting this mutation is 50-50, like the autosomal dominant, like we mentioned. She could either 50% chance she has this mutation, 50% chance she doesn't. And so we will test her for it. We will test her for the mutation to see if she has inherited it or not. But let's say that instead of a SOD1 positive result, we did a panel test on mom, and mom came back negative. So we did not find any change in mom's genes that could have led to this ALS. Then in this case, we can tell that something is going on in this family, because there's 4 people in this family who have had ALS, but we couldn't pinpoint a particular genetic mutation. So maybe this falls into that 20% of people with unknown changes in their DNA, which we're not able to figure out yet. But in that case, for this person, genetic testing really isn't indicated, because we don't have anything to test them for yet. We would just ask mom to keep following up, and maybe eventually, when there's more genes that we know of, get retested in hopefully 5 to 10 years when there's more known. But there would be no purpose of testing this unaffected member then if the mom came back negative. And then the last scenario is, and this is a scenario that we see very commonly in neurological conditions, that all affected family members have passed away from the condition. And this person, let's say this person, that really limits your options, and you're only left with the unaffected person left to test. But let's say this person got tested for a panel and came back positive for the SOD1 mutation. So then we would assume that mom, brother, and everybody else also likely had the SOD1 mutation. But if they did a panel test and came back negative, that doesn't tell us about the risk for other family members, because it could be that mom had a SOD1 mutation, but just didn't pass it down to this person. Maybe she could have passed it down to the other sister. So really, that's what I meant by true negative. This isn't a true negative, because if this is a true negative, if mom was positive and didn't pass it down, then we know that this person is not at an increased risk of developing ALS. But it is really a complicated and frustrating position for a lot of unaffected people to be in, where they don't know their risk sometimes. Great. I think that was all I had for this slide. And I just wanted to end by saying that, like I said, genetic testing is a really complex thing to go through in general, and genetic counselors and your neurologists are usually going to be there for you to walk you through the process, and you might need them to, because there's a lot of hanging parts with genetic testing. But if anyone has any questions, I would be happy to answer them.
Okay, Shrika, I'm going to reclaim, and then yes, we have a couple questions.
I don't think I can see them. Maybe I should stop sharing. Before I stop sharing, I have my last slide is a slide of resources. It's basically all of these resources are things that I use to make the slides, and they're pretty patient friendly as well. There's also a link to finding a genetic counselor. So if anyone is interested in genetic testing or knows that there's a specific change in their family, you can go to this link, put in your zip code, and they'll show you genetic counselors around you.
Great.
Okay. I can.
Shrika, that was a lot of great information.
Thank you.
Thank you.
I have a question. You talked a couple times about testing the affected person.
Mm-hmm.
So what if the affected person is over 74?
Mm-hmm.
Right.
Since they don't recommend genetic testing for persons over 74.
I think it's a lot. I think for a lot of these conditions, we still would recommend testing that person first, regardless of their age, because, like I said, it's really hard to get to understand what the unaffected person's risk will be without testing the affected person, because really that affected person is going to be the most informative person you can test in the family, because if they come back negative, you can know that if there was going to be a genetic change in the family, the affected person was going to have it. So if the affected person comes back negative, you can be sure that you've tested everything that you possibly can, and there's nothing identified right now. But if the unaffected person gets tested, you're really like, maybe this person didn't inherit the change in the first place. So it's really not as informative as testing an unaffected person. So I wasn't aware of the 74-year-old cutoff, because I think we would still recommend test. It's a simple blood test. It really isn't much. It really isn't that strenuous on people. So I think we would still recommend testing, right, Lauren? Yes.
That may have a bit to do with insurance coverage.
Oh, okay.
Mm-hmm.
Got it.
So that may be something that if family members wanted to do that, that may just be an investment that they would have to make on their own.
Right. And genetic testing, I know a lot of people view it as something that's very, very expensive, because it used to be very, very expensive. It is much more accessible nowadays. A lot of labs do testing at really affordable prices, between like $250 to $500 out of full self-pay options. So really genetic testing is more accessible now than it used to be.
Right. We have opened the chat for questions. If anyone has any questions, we can ask Shrika now. Well, Shrika, you gave a lot of great information, so you were very thorough. That may be why we don't have questions.
That's fine. And if someone has questions at a later time, I'm sure that Lauren and I would be happy to answer them through email.
We just appreciate your time and the continued collaboration with the Emory Genetic Counseling Program.
Of course, yeah.
Well, thank you for having us, and have a healthy and safe holiday season.
Great, thank you so much. And we look forward to our next genetic counseling webinar, which will be with Jay
December 15th.
on December 15th.
Excellent. Well, thank you, everyone.
Thank you.
Thank you, Shrika. Excellent job.
Sharika, thank you.